So hi everyone, and a warm welcome back to the Pelago Bioscience webinar series directly from our studio here in Stockholm. My name is Helena Almqvist and I'm a Senior Product Advisor here at Pelago, and with me I have my colleague Lawrence.
Hi, I'm Lawrence, Head of R&D. It's a pleasure to be here to assist with questions from our audience today, so please submit them via the webinar's Q&A function and we'll take them at the end of the seminar.
Great, thank you Lawrence. So it's now time to introduce our speaker for today, Jon Ellery. Jon is currently Director of Biology at Sitryx. Before that, he has worked in multiple companies, for example, Takeda. A key focus for Jon, though, has always been to use analytical pharmacology to guide development of small molecules and maximizing confidence in the translation into the clinic. And that is exactly what we're going to hear about today. So the topic of today's webinar is how CETSA has been applied to translational studies to bridge the gap between in vitro and in vivo. So with that, I'll leave the stage over to you, to Jon.
Thank you, Helena, for that kind introduction. I am delighted to have the opportunity to participate in this webinar, where I can talk to you about how Sitryx has worked with Pelago to deploy CETSA and the impact it has had on one of our preclinical drug discovery projects. The title of the webinar is CETSA, a translational tool. In this webinar, I would like to cover the following topics: a brief introduction to Sitryx, some background to the preclinical project I will be talking about. The majority of the webinar, though, will focus on how we first used CETSA to show compound engagement in an in vitro system, and then how this was then adapted to demonstrate target engagement in tissue from an in vivo model. Let us start with a brief introduction to Sitryx. Sitryx was founded in 2017 as a small molecule drug discovery company based in Oxford, UK. Our focus is in the field of immunometabolism, which I will explain briefly in the next slide. As a company, we are dedicated to bringing novel therapeutics to the clinic that address disorders of the immune system for the benefit of patients. We work closely with our partner organizations to drive our projects forward in a data-led manner, seeking out the appropriate expertise as we progress through this endeavor. So in this, we have been, to date, fairly successful, with a molecule in clinic in partnership with Lilly, possibly going to phase 2 this year, a molecule with a novel mechanism toward PKM2 modulation, due to enter clinic this year for atopic dermatitis, a first-in-class inhibitor of MTHFR2 on track to enter the clinic in 2026 for psoriatic arthritis. Due to the mechanisms of these molecules, they have the potential to address additional complex immune disorders that would bring potential benefit to more patients. So we can continue to build our understanding on these targets for that purpose. Now, behind this, we have built a strong internal team with a growing understanding of how this biology works in practice, and subsequently how to translate this into the delivery of clinical candidates in this field. As you can see, we are backed by a strong set of very supportive investors. So what is immunometabolism? Firstly, it can be regarded as a precision immunology therapeutic opportunity. The metabolism of immune cells changes in response to stimuli. A naive, unstimulated immune cell is not proliferating or secreting soluble factors. Stimulate that cell, and then it begins to divide and to express a whole host of molecules. It has woken up, and its metabolism needs to power up to cope with the new demands. In many diseases that involve the immune system, we can see profound and sustained changes in cellular metabolism of those immune cells. For example, on the left of this slide, we can see, looking at the green dye, you see the localized increase in glucose metabolism in psoriatic arthritic lesions. That is due to activated immune cells. We also know that when activated in a certain context and environment, the immune cells upregulate metabolic pathways and manifest particular phenotypes, as shown on the right-hand panel. The metabolic profile adopted depends on the cell type and the integrated external stimuli that it is receiving. So, for example, a macrophage will have a different profile to T helper cells or a B cell, even if they are in the same tissue environment. The two are directly linked, meaning that if you alter the metabolic pathways in the immune cell, you can alter its phenotype. So in many diseases where there is an immune component, certain immune cells present a particular phenotype that is damaging. Now, these may predominate, and initially they may have been present as part of a completely appropriate immune response, but for some reason, they have become stuck in that particular phenotype and activity, and eventually that has become non-beneficial in a chronic setting. In the field of immunometabolism, we aim to modulate the metabolism of dysregulated immune cells to alter their phenotype from one associated with the etiology of a disease to a beneficial state. For example, in pushing an inflammatory macrophage towards a non-inflammatory resolving phenotype. We have done this by working closely with other curious and data-driven organizations across the scientific community, both academic and industrial, to bring molecules to the clinic to understand the therapeutic benefit of this approach for patients in not only improving treatment options, but improving their quality of life. In this way, we've been successful in moving several compounds into the clinic, as I mentioned before. Sitryx is therefore a leader in the field of precision immunology therapeutics. We have a broad pipeline that is beginning to enter the clinic. There is a strong, seasoned team of drug discovery professionals in our leadership team that have a track record of delivery. And with our clinical assets, we are looking in the near future to gain an understanding of the real therapeutic potential that immunometabolism has on complex diseases with an immune component. Let us now look at the background of the project that will be the subject of this webinar. The target is an intracellular enzyme. It has two isoforms, one residing in the cytoplasm and one in the mitochondria. We believe it plays a central role in directing the flux of metabolites within the cell in response to external stimuli. We observed that there is an upregulation of enzyme activity in activated macrophages, providing key intermediates for the production of pro-inflammatory cytokines. There is also enzyme upregulation in differentiated fibroblasts, supporting the production of extracellular matrix proteins. Idiopathic pulmonary fibrosis, or IPF, is a chronic, progressive disease with a very poor prognosis and a severe impact upon people's quality of life. When I mean poor prognosis, the survival rate is roughly from 4 years from diagnosis. This is mainly driven through the rapid and progressive loss of lung function due to the deposition of fibrotic material. The result is breathlessness, loss of mobility, and inability to do even simple tasks at the later stages of the disease. And people often require additional oxygen at these stages. It is very poorly addressed clinically, with lung transplantation being seen as one of the few options, in the late stage. Therefore, there is plenty of opportunity to develop compounds that would provide significant therapeutic benefit and improve patient quality of life by an enormous measure. Now, pleasingly, recent clinical trials by others are showing promising results, and this is pleasing to anyone in this industry that we are having an effect on this horrendous disease. In patients with IPF, we see that there is an upregulation or dysregulation of the target enzyme activity, especially in the fibrotic active areas of the lung. We hypothesize that inhibiting the enzyme in the lung of IPF patients would reduce inflammation and the deposition of fibrotic material. It is important to note that we believed that we would need to inhibit both isoforms, or better, that we did not have any evidence that only inhibiting one isoform would be beneficial. And so our approach would target the active site that was identical in both isoforms. Additionally, we wanted to understand if inhibition of this enzyme in resident macrophages could cause them to switch to a resolving phenotype, with the possibility of helping to reverse tissue damage and improving lung function. This would represent a major improvement for patients in addressing this disease. At the beginning of the project, we defined our target compound profile. We wanted to target the lung, so opted to deliver the compound via an inhaled route of administration. The challenge here is to deliver enough compound to the extremities of the lung, as illustrated in the diagram. You can see that you inhale at the top of the airway, and you need to be able to deliver enough compound through all the intricacies of the ever decreasing size of airway down into the extremities of those lungs. So to achieve this, we needed a very selective and potent compound against both isoforms. It needed to be highly soluble with low oral bioavailability, as a lot of any inhaled drug ends up being swallowed and entering the gut. It needed, though, to have reasonable permeability, so compound that did get into the right location within the lung could be absorbed. To aid with low bioavailability, we wanted low hepatic stability. So any compound being absorbed or getting into the circulation would be quickly cleared by the liver, and therefore really limit the potential for systemic exposure. Now, this does contrast with most small molecule drug discovery, where we are looking to have oral bioavailability and thus want high hepatic stability. And it took us a little bit of time to get our head around that fact. So in contrast, though, we wanted it to be stable in the lung. So it needed to have high stability in the S9 fraction of lung tissue, where most of any metabolic enzymes tend to reside when fractionated. And we were therefore aiming for a half-life in the lung of greater than 4 hours. Obviously, it needed to pass all the normal safety criteria, and in addition, being inhaled, we needed to check for irritation in the airway and a suitable vehicle for this route of administration. Now, we have to check for the irritation because you normally have a very high concentration of compound when you are dosing, and just that high concentration of compound can cause irritation independent of any pharmacology of the compound. Let us move now on to how we deployed CETSA to help move this project forward. Firstly, using a very robust design make test cycle, or DMT cycle, our medicinal chemists were able to design very potent compounds with great selectivity, as seen here with SYX0000XX. Apologies, I will not be able to disclose the actual compound numbers of our own compounds in this webinar. This compound inhibited the recombinant enzyme with a PIC50 of 7.3, that is 54 nanomolar, and shows a PIC50 of 8.7, that is 1.8 nanomolar, in a cellular assay. It also showed no activity towards a closely related enzyme. We were able to use just one isoform in the DMT cycle because the target binding site was identical in both isoforms. And our cellular assay consistently showed this greater sensitivity towards inhibitors, regardless of the chemotype that we were testing. It is worth mentioning at this stage, we did not know if the response being observed in the cellular assay was due to the inhibition of one or both isoforms. The key question being whether our compounds could also cross into the mitochondria. Anecdotally, there are suggestions that compounds need certain properties to efficiently penetrate the mitochondria and engage with targets in that organelle. In addition to the great potency and selectivity, we're designed in the desirable properties required for an inhaled drug as we had previously defined. High solubility, low hepatic stability, but stable in lung S9 assays. It had reasonable permeability, and we were able to add in basicity that has been shown to help enhance retention of compounds in the lung. Owing to the nature of administration and the target tissue, we decided that we needed to be able to show target engagement. We opted to see if CETSA could be used. So what is CETSA? So CETSA, which stands for Cellular Thermal Shift Assay, is a methodology for looking at how compounds can affect the thermal stability of a protein in the complex environment of the cell or tissue. In a given biological environment, as the temperature is increased, a protein will become denatured. The denatured protein can aggregate, and by spinning or by some other method, the denatured protein can be removed from the suspension, and the percentage of some remaining soluble protein can be determined. And this can be plotted against temperature to form a melt curve, as illustrated by the left curve in the diagram on this slide. That is the blue line. A compound that binds to the protein target can stabilize the protein in a way that it means it denatures at a higher temperature. And this can result in a measurable rightward shift in the melt curve, as illustrated by the red line of the diagram above. The magnitude of this shift is dependent on the target protein and how effective the compound is in stabilizing it, which may be a consequence of its binding modality and location of binding. At the time of this work, we used a specific antibody to monitor the level of protein remaining in solution. It is critically important that you can accurately identify and follow your protein of interest. As we all know, the specificity of antibodies, certainly commercial antibodies for research, can be problematic, and Western blotting can show a degree of variability as well. Pelago has enhanced the CETSA methodology further by introducing a mass spectrometry approach that relies on detecting peptides specific to the target protein. And this has expanded the utility of this technology to not only looking at specific target proteins, but how other proteins in a cell may also be affected, either destabilized or stabilized because of the actions or because of your compound actions on your target protein. This is not the subject of this webinar, but I'm sure the team at Pelago will be more than happy to talk to you about it. Before looking at target engagement in vivo, we wanted to understand if the target was amenable to CETSA. So we needed to validate the technology in an in vitro system first. As I mentioned before, we did not know if our cellular assay data was driven by engagement of one or both of the enzyme isoforms. We therefore also wanted to use CETSA to understand whether compounds were able to penetrate the mitochondria and stabilize the mitochondrial isoform as well. We treated a cell line known to express both isoforms with either a set concentration of a tool compound, which was known to be able to inhibit both isoforms, here labeled SYX003724, or with DMSO. We then split the cells into equal aliquots, heated aliquots to specific temperatures, isolated the soluble fraction, and detected the soluble protein by Western blot. Normally, you plot the data as a ratio of signal relative to untreated control at a set temperature where there is no denaturation observed, here chosen as 43 degrees centigrade. This is then plotted to form melt curves plus and minus compound. As you can see, in the presence of 0.25 micromolar tool compound, we have a rightward shift in the melt curve. That is, pink line moving to turquoise line. This is roughly 10 degrees centigrade on both isoforms, which is a very big shift. Just to make you aware, shifts that are observed are normally smaller than this. And for your given target and compound combination, you would need to establish the robustness of the response and whether it is reliable. Now, if you wanted to compare the concentration-dependent stabilization activity of different compounds to rank them against a target protein, you pick a set temperature at which to perform the experiments. Using the data above, we chose 49 degrees centigrade as the set temperature at which to assess the concentration effect of compound on protein stabilization, because we have a small, consistent amount of soluble protein in the absence of compound that acts as a great baseline against which we can assess the stabilization of the protein in the presence of compound and to define maybe quality parameters for the assay as well. So using this approach, we tested a number of compounds which we could then rank in terms of their ability to stabilize the target protein. Here we can see that the exemplar compound shown previously in the recombinant enzyme and cellular assay stabilized both target isoforms with a similar EC50 of 35 nanomolar and 55 nanomolar respectively. This gave us the first clear indication that our compound could engage both isoforms and was therefore able to not only cross the plasma membrane, but was also able to cross into the mitochondria. This information could be fed back into our understanding around compounds and help inform further design. It also gave us great confidence to move forward to see if we could use this approach to ask questions around target engagement within the lung tissue. So the next section focuses on how we subsequently use CETSA to look at target engagement in vivo. Firstly, we wanted to make sure that we could harvest and process lungs in a manner that allowed the antibody to not only detect the target protein, but that it could be stabilized by our compound in a similar manner to the in vitro experiments. As we are looking at a single compound in each experiment, it was decided that generating melt curves would be the most appropriate route forward when looking at target engagement within a tissue. So here you can see that we took healthy rodents, removed and processed the lungs before adding either compound or vehicle. The Western blot demonstrates that we could process the tissue to obtain soluble target protein that was sensitive to denaturation by increasing temperature, and that compound could stabilize the target protein in the processed lung tissue. You can also see that we get a clearer distinction in the denaturation if we look at the lysate rather than the homogenate. Thus, using CETSA, we confirm that both isoforms of the enzyme target were expressed in the lung tissue at detectable levels, and that after processing could be stabilized by binding compound. I'll have to say now, it's not a trivial undertaking to optimize this process. And we were very impressed with how the team at Pelago worked through the optimization parameters to land on a robust methodology for us. Next, we asked the question whether if dosing the animals before harvesting and processing the lungs as optimized previously, we would still observe stabilization of the target protein. There was no guarantee that during processing, any bound compound would not just dissociate and be lost. So we gave animals a high dose of compound via the inhaled route. We then harvested the lungs after a specific time and processed them as before. You can see in the Western blot that in an animal dosed with compound, that we retain bands on the Western blot at higher temperatures than the control animal dosed purely with the vehicle. This is very important because not only are we seeing engagement of both isoforms, we retain that engagement equally, and therefore this is unlikely that the mitochondrial isoform is being bound by a compound that has been released during the process. By graphing the data as a ratio, we could clearly see a rightward shift in the Melt curve for both isoforms. There is admittedly more variability behind this combined data due to the inherent variability associated with animal models and with an inhaled route of administration, and the resultant differences are distribution within the lung of those animals. This, though, was our first real evidence that when we dosed animals by an inhaled route, that the compound could get into the lung and engage with our target enzyme isoforms. Although we had observed target engagement, we performed a mass spec analysis of the product and substrate levels of the enzyme to ask if there was actually a functional consequence on enzyme activity. In this, we determined the ratio of substrate over product in lung tissue as a way to account for the natural variance observed among animals. Now, this is further complicated because we know that for this product and substrate, there are other metabolic processes that could generate them. The increase in ratio indicated by the pink dots indicate that we had less product being formed and could thus infer that we were seeing a functional inhibition of the enzyme. This increased our confidence enormously and allowed us to begin to ask other questions, such as, was the inhibition observed because all of the enzyme present in the tissue was inhibited by the compound? The question of how much of the target is engaged is an important question that we need to ask, as it helps in understanding the pharmacodynamics of a drug. If you only need 10% of your target to be bound with compound to have the desired beneficial effect, you could potentially need to use a smaller dose and reduce the chance of side effects. So, how did we look at this? So, in conjunction with Pelago, we determined that we could look at it in the following way. Take a set of processed tissues taken from animals that had either been dosed with compound or vehicle. Divide each sample equally to form two groups. To one group add more compound, and to the other vehicle, as in DMSO. Generate the Melt curve. The thinking here is that the added compound would bind any free protein and stabilize it, shifting the level of observed stabilized target protein and revealing any difference with tissue where no extra compound had been added after processing. Looking at the Western blot, there did not seem to be any obvious difference. And when the data was normalized and plotted, we could see that there was no obvious difference between control tissue and dosed tissue with added compound and dosed tissue alone. This is a clear indication that there was no significant proportion of protein in the samples taken that was free to be stabilized by additional compound, and that we had near 100% target engagement. It also infers that the compound being administered by an inhaled route had not only really penetrated into the lung tissue and managed to engage target protein wherever it was in that tissue. We also wanted to understand how target engagement changed over time post-dosing. Again, this is important as it will help inform our understanding of the pharmacodynamics of any efficacy that we are seeing, especially in relation to the pharmacokinetics of the compound. The key difference in this study was the time after dosing in which we harvested the lungs. We chose 3 time points, 5 minutes, 8 hours, and 24 hours, at which to harvest the lungs and to perform CETSA. We also took samples for PK analysis, ensuring that we had samples taken at the same time points as harvesting. Western blot showed there was stabilization and it possibly changed over time. So we proceeded with analyzing the data. If we look at the right-hand graph, we can see a PK plot for SYX 0000XX. The solid line shows the concentration of unbound compound in the plasma. It indicates that we have very low plasma and hence systemic exposure, and after 4 hours was below the level of quantification. It also shows with the dashed line that initially there was a very high concentration in the lung. This is expected because of the high dose when administering compound by an inhaled route. There is an initial rapid decline as compound that is probably trapped in the lung mucus is removed and swallowed. And after about 1 hour, there is then a steady decline over time. Thinking back to our target compound profile, this is the type of profile that we were aiming to achieve. Previously, we had defined by Melt curve an in vitro EC50 of 35 and 55 nanomolar respectively for the 2 isoforms. Referring to this data, we observe that the unbound concentration of SYX 0000XX in the lung is around this level by 8 hours post-dosing and below that level by 24 hours. Now, looking at the CETSA data on the left for isoform 1, and we are only showing one isoform for clarity, though isoform 2 shows the same pattern, we can infer that the target is fully engaged at 5 minutes and 8 hours post-dosing, whilst by 24 hours we can see that there is a decay in stabilization, indicating a loss of target protein engagement. This tells us that there is a very rapid penetration of compound into the lung and a rapid target engagement of compound with the target in the lung as well. Now, because at 24 hours, the PK is indicating the compound concentration to have fallen below the Melt curve EC50, we can hypothesize that we have the potential for a small disconnect between target engagement and free compound concentrations, because there is still some stabilization occurring. It is to be mentioned here that we do not understand where the rate of protein turnover of this target within the lung at this time. So some of the loss of target engagement may be due to degradation of protein as well as replacement with new de novo synthesized protein. If we wanted, this could be further assessed using a range of different doses to build out a comprehensive data set of dose, exposure, and target engagement over time. You can imagine that such data would be very useful in working towards an understanding of what is important in predicting human dose, especially if the samples were being obtained from animals in an efficacy study in a disease model. So let's summarize what I've spoken about in this webinar. Hopefully, I've shown you the following. Sitryx has developed potent and selective small molecule inhibitors to an intracellular target that we believe plays an important role in the disease mechanisms behind IPF. We have shown target engagement in both an in vitro and an in vivo setting using CETSA, showing that our compound can be effectively administered via an inhaled route and that this equates with inhibition of target enzyme activity. We have demonstrated that near 100% target engagement can be achieved and the level of target engagement can be monitored over time. This current project is still in preclinical development, but making steady progress towards its desired endpoints. Finally, but most importantly, I'd like to acknowledge the great team at both Sitryx and Pelago that work closely together as one team to develop the assays and work through the optimization of the in vivo experiments to drive this work forward. It is a testament to the curiosity and positive approach to overcoming technical challenges in both companies that has resulted in such a beneficial collaboration. And with that, I'd like to thank you for your time today, and I am happy to take any questions.
So thank you very much, John, for that very insightful talk. And so now it's really time for the Q&A session to start. And me and Lawrence, we have prepared a few questions. And if you do, please so write questions in the chat, and he's happy to answer your questions as well. But let's go ahead and start the Q&A session. So Lawrence, go ahead.
Yeah, thanks. A really interesting talk. So in your study, you discovered that the applied dosing resulted in a maximum or saturated target engagement. And coming at this from another angle, would you consider the application of CETSA to set the dose in animal studies from the cell-based data going forward?
That's a really interesting question, Lawrence. Hopefully you can hear me as well. So working using CETSA first to then define the dose. I think what we'd have to do is, like anything, we'd like to build up our correlation. So you might have noticed we used this 2 ml per kg for 5 minutes, and essentially we're getting near 100% target engagement in our assessment. We didn't really do the same level of looking in terms of in the in vitro setting and trying to match up what we saw in the cell-based system with the sort of the potential inhibition effect that we were seeing. Now that could have actually been really interesting, because we were assuming that just using the top dose we could get this lovely curve and we could see the melt and show target engagement. Actually, it's a really good point, I think, that doing essentially a target engagement and then backtracking to see, at least in a cellular system, do you still need 100% to track? Does it track with the IC50 of the functional response? That's a really good point, actually, because that could have helped immediately start saying to us, you don't necessarily need to get 100% in the lung tissue, which is very important. I know I mentioned about the irritancy, which is predominantly driven by the huge dose you give in a very short space of time into the lung. It's like breathing in lots of dust, basically. It makes you choke and stuff like that. Obviously, that's not how we dose. But if the lower the amount of compound you can put in and still get the benefit, the better it is for you. So that's a really interesting way of looking at it, Lawrence. And that's something maybe to think about going forward for these quite a little bit more challenging dosing routes that we sometimes do. Yeah.
Okay, so moving on to another question. You mentioned that you had very selective compounds. So I'm just curious, this is a kind of a two-part question. So first of all, how did you assess selectivity? What panels? I guess you used some standard panels. But then sort of the second part of that question is that since you described that CETSA and we can do it in protein-wide to assess selectivity basically in any matrix. So elaborate a little bit about the potential value of actually do it in vivo to cross-check and correlate the selectivity also in the in vivo setting. So what do you think about that?
Again, a great question. So like many small molecule companies, we run the standard panels. We go to Eurofins, you might call it the SERAP panels, the safety 44s, the cardiac ion channel panels. Everyone's used to those. So that falls into your standard category. Now, when we were starting out the CETSA with Pelago, you mentioned we were using the antibody approach. We were a little bit early for the mass spec approach, and the proteome was coming out a little bit later than that. Given you weigh up the costs, etc., if this was that was our lead compound, I think it would have been incredibly powerful to then move forward and test with that route. What else are we modifying? A proteome-wide assessment in actual tissue is very powerful, just to confirm that if you do see some unexpected results, for example, that you might even be able to identify which protein is being stabilized, and therefore is there an interaction that you did not know about, i.e. with the target and another protein which is peculiar to that tissue, or whether there's just something you haven't been able to check, because to be honest, we can't check every single protein. we'd be here forever if we're having to check every single protein. But being able to do it in a tissue, I think would be incredibly powerful. So certainly, I think if you see something that you're not expecting, you might follow that up. Whether you do it if just for completeness, I'm not sure. I think you'd have to assess that on the risk basis each time. You know, sometimes we're reluctant to generate data on our lead compounds, which becomes difficult maybe to explain.
Yeah.
You know?
Yeah. So then just to add on that, so the CETSA protein-wide can have sort of potential new targets that you don't have in the standard panel. So yeah. But great question. I think we have some questions in the chat here actually. So
we do have a couple of questions here. So in your in vivo compound dosing and subsequent CETSA readout, did you observe dose dependence of these effects? And could you correlate these with dose dependent effects on a PD endpoint to define PK/PD understanding?
A really great question. Now, obviously, I haven't disclosed that in this particular webinar. That would have been a little bit longer, but we were seeing PD effects. Now, we all normally go away and pick out some arbitrary in vitro IC50 value, that we've come from an enzyme assay or a cellular assay, and just then try to correlate that through to our to what's happening in vivo. That's great if it's linear and it works dose and you can work out your free exposures and everything's fine. But when it's not necessarily a linear relationship, we found that the CETSA is really useful to actually then look at, actually, is it the target engagement, which is the really important thing? Because we know the in vitro situations are, they're so sort of false. They're not really physiological. You're really defining conditions very precisely to give you a maximum readout. Whilst if you look at the tissue, everything is in there. Everything that possibly might want to interact or is important for the PD readout is playing in. And you don't know how it amplifies through. So you may need end up only needing 10% of target engagement to still get the same effect. And again, that follows through to the dosing of the lower the dose for an inhaled route you can get and still have really great effect. That is where you're driving to, because it just, like anything, we all know that the lower the dose we can get, we have fewer chance of an off-target happening. So, really, hopefully that answers the question. So we did see a PD correlation, and the CETSA did help us understand what was driving that.
We have another question here on the dosing theme. Have you tried measuring in vivo target engagement with a lower dose to observe an added TE response or a target engagement response with an ex vivo supplementation?
Okay, I'm trying to get my head around the question. That's quite a complicated question, I think, here. So measuring, so is this a little bit like the target engagement where we're seeing the loss of target engagement, say, at 24 hours and then following up? Yes. So, I didn't show that data, but we did follow that up. So where we had confidence we weren't seeing 100% target engagement, yes, we could then add extra compound ex vivo and push that up to the 100% level. Hopefully that was answering the question that I think I heard.
That was my understanding, so yes, I think so.
Okay. And in fact, one of the curves on that graph is actually target is tissue which hasn't been exposed to compound at all. It's just been exposed to the vehicle and then had the compound added. So that was our positive control to bring it up to the 100%. So,
I'm jumping to a sort of a non-scientific questions. I mean, you probably work with more CROs than Pelago. So I'm just curious to hear what is important for you when working with the CRO.
I think great communication is the most important thing here. What I've always found is if you have an open dialogue with people and you are incredibly curious about the data. And that's the critical thing, I think, for any good CRO. I know that there are pressures on making your own profit and turning around the data. But what we'd really like to, at least with Sitryx tries to work as one team. It's part of our values that we work as one team, not just within our organization, but with our partners. We like to regard our CROs as partners. And we like to include them in a very open and transparent communication about what we're doing, because we feel that we regard internally that we're really great scientists. Who doesn't? But we also recognize that every CRO we work with is also full of really passionate and great scientists. And why not capitalize upon that? Why not use and engage that resource and that excellent mindset to help build really robust assays? And what you've seen today in this webinar is some of the challenges that we faced at the time. They were not necessarily the standard protocol with Pelago. So we were working hand in hand with Pelago and the Pelago scientists to work out how we could identify whether we had 100% target engagement. How do we work out whether we can even see the compound binding in the tissue when it's being dosed pre-harvest? Because there's no guarantee that the compound will not just dissociate and disappear when in the actual processing. So we really appreciated working closely with the Pelago scientists, having that open dialogue, that great communication, and the regular updates, and just being aware of what was going on, and both sides being open to suggestion and curiosity about how things happen, how things work. So rather than sometimes this is the way it's done, it was very much of, well, okay, that's how we start off, but how could we do something different? What's appropriate for this particular protein and this project? And as you can see, that worked really well.
Yeah, thank you, Jon. We can just say the same. It was a great collaboration.
Did have a further question on in your talk. You showed a literature compound that had a very nice target engagement in both the cytosolic and mitochondrial isoforms. And you were talking about anecdotally passing that mitochondrial membrane. I wonder, did you learn anything about the chemistry required to do so?
This is a really great question, Lawrence. So depending upon who you listen to in the chemistry world, in the field of chemistry, and again, these are really intelligent people who've worked on multiple projects and had multiple challenges. There's one school of thought which says there are specific requirements for the compound to have, so phys-chem requirements for that compound to not just penetrate the plasma membrane, but to then successfully penetrate the mitochondrial membrane, because that mitochondrial membrane is different. It's not just the plasma membrane within the cell. It's a mitochondrial membrane. It's different. And we did have one project where we had exactly this. The chemistry was very exacting. It didn't seem to be whatever we did. Only very narrow physical chemical properties would allow those compounds to enter the mitochondria. For this particular project, though, in true fashion, we were quite happy to stand up in front of our colleagues and say, well, it doesn't seem to affect our compounds at all. They seem to sail through. So I probably would understand that it's a combination of your core chemistry and your particular target, and where it might be located within the mitochondria, rather than just per se, it's a one size fits all. Certain chemotypes will sail across, other chemotypes won't. Whether they then end up in the right part of the mitochondrial matrix, or to be able to engage where your target is a different question. So I would say it's a lot more complicated than people think. Yeah. But we were lucky, basically, I think.
Luck is always good.
Yeah,
we have more
luck's good,
questions.
isn't
Yeah, we have more questions in the chat, and so here I found interesting that your target engagement was decreasing over time, 5 to 8 minutes to 24 hours. I suppose it would depend on the drug and the target, but what is your opinion on detecting target engagement after long-term dosing of the drug to mice?
So that particular piece was done after just a single dose, and we were following. So you could see from the PK that the free fraction was decreasing with time, as you would expect, as a compound is sort of essentially removed from the system. What we didn't do is we then didn't redose. So we didn't follow a multi-dose regime to see whether we could then, increase target engagement again after. We were just interested to try and understand if there was a real disconnect between the PK and the sort of target engagement there, because sometimes you put a compound on, and its off rate is so slow that it's potentially covalent, and for some targets that's actually a really good thing. You can, again, we were looking potentially say, oh, do we need to dose every day, or could it be every other day, or would it need to be every 12 hours? And you can see from that CETSA data, we're getting an indication that, okay, maybe it would have to be every 24 hours. But we were also seeing that the target engagement was not necessarily completely tracking with the actual PK. So we could have upped the dose a little bit, or what we haven't obviously disclosed is how much target engagement we needed to have the effect. We were actually probably looking more that we could dose every other day at that dose level, meaning that we could potentially have got in with a lower dose, again, looking at that, reducing the chance of irritancy and off-target effects, and then dosed every day, which is probably easier for patients to get their heads around rather than one day on, one day off. It can be a little bit, if you forget, then you're completely out of sync. So again, that was starting to give us that understanding of, can we get away with a lower dose, and how often will we need to dose? Yeah, it's a great question though, because ideally, when you get to that, getting closer and closer to your candidate, you'd probably then say, okay, let's see if we can get away with a lower dose, build that correlation. Where do we need to go to get that target engagement for that length of time, and how many doses do we need per day? Hopefully one, etc. So it is a very good question. Yeah.
Okay, great. So we have one more question in chat, and I think in the interest of time, we take that question and then we wrap up. But maybe Lawrence, if you want to take that question.
Have you tried or planned to use CETSA with samples from the in vivo lung fibrosis models or in vivo mouse lung fibrosis models dosed with your compound?
So this is a really great question. Now, this might depend upon whether you believe the mouse lung fibrosis models are actually worth doing in the first place. So what you've seen here is we're looking predominantly at the healthy lung, and we were looking to see about how well the compound penetrated the lung and managed to engage the target. We would be, if we are still pursuing this particular avenue and looking for that fibrotic material, we would definitely be looking to use CETSA there, because that, I can understand this question. It's a really important one. Is your target still expressed actually in the disease tissue itself? So we would be isolating maybe the fibrotic material on its own versus maybe the non-fibrotic material closer, further up the lung, and actually doing the comparisons. And it's a really important point, that, because then you're building that confidence that it is actually engaging the fibrotic material. Ideally, of course, in our hypothesis, we are actually hoping to basically reduce the amount of fibrotic material. So that would be a very long study. And as it is, unfortunately, in this particular format, quite terminal to the mice, we're not entirely sure at this particular point how much more you would gain from it. Because if you've managed to reduce the amount of fibrotic material compared to your wild type, you are confirming that is the key indicator there, that you're reducing fibrosis overall. The CETSA is like the icing on the cake, that yes, it's still engaged. I'm not sure it would be the driver at that point, because It's one of those, if you're talking about something like a bleomycin model, everyone wants to see it, and then everybody tells you that no one believes it. So it's a bit of a difficult one.
Thank you, John. The icing on the cake, that's a good wrap, I think. So thank you very much, John. Thank you very much for everyone attending. This was super fun. We'll do it again, so stay tuned for our next webinar. Thank you very much.
Excellent. Pleasure. Thank you, everyone.